Hello everyone, welcome to all of you. So in last lecture we have discussed about pistil and megasporangium, structure of ovule and type of ovule. We have already gone through it, right? So in this lecture we are going to discuss about megasporogenesis, right? So megasporogenesis, it is the process of formation of megaspore from the megaspore mother cell, MMC. It is known as a megasporogenesis. Why it is called megaspore? Because female gamete it is larger than the male gamete. That's the reason it is megaspore. And process of formation of this megaspore from the megaspore mother cell, it is known as a megasporogenesis. Genesis means formation, right? So ovule generally difference in a single megaspore mother cell, it is MMC from the nucellus. You can see here we have already gone through this is nucellus, right? So there are so many cells in a nucellus. We can see here, you can also see there are so many cells in nucellus. So one of cell of this nucellus will be behave as a MMC, megaspore mother cell, right? So this megaspore mother cell, or we can say it is MMC. So this MMC will undergoes in meiotic division, right? So we will be having, this is first meiotic division, and there will be a second meiotic division, meiosis 2. So we'll be having 4 megaspore tetrad, and one of these 4 it will be remain activate, and 3 of these 4 will be degenerate or deactivated, right? So that's the reason this this embryonic cell sac also called as a monosporic embryo. Why? Because from only one megaspore, whole embryonic sac it is forming. That's the reason this embryonic sac also known as a monosporic. Mono means once. So this is haploid, right? Because it is product of meiosis. That's the reason these all are haploid. So 3 of these 4 have already degenerate and 1 remain active. So from this one it will be undergo from the mitotic division, right? So we'll be having 2 nuclei. So first there will be a karyokinesis. Now what I mean by karyokinesis? It is nuclear division, right? So this is first mitotic division one time. This is second time mitotic division. So we'll be having 4 nucleus, right? So from this one we will be having 2. So after second term, and this is third time mitotic division. That's the reason we will be having 8 nucleus. You can see here this is 4 and this is 4, so we'll be having 8 nucleus. So you can see here this is micropillar end, pilar end, and this is chalazal end. So 3 will go at this micropillar end, and 3 will go at this chalazar end. So you can say, now I'm converting this structure, right? So don't get confused in this. I'm just invert this structure. So you can see here this is chalazal, now this is chalazal, and this is micropillar. So now each and every nucleus will form cell walls around it. So this is second cell wall and this is third cell wall. So you can see here this is antipodal cell, and they are 3 in number, right? So and this is polar nuclei. So it is not forming any cell wall surround it. And this is egg apparatus. Egg apparatus, it is formed by egg cell plus synergid. Now synergid has a very beautiful function. It will work as an indicator. It will indicate pollen tube, and it will show where to the pollen tube, like from where it has to be entered. So and by this as I told you earlier, this is egg cell and plus synergid, it is forming egg apparatus. So egg apparatus having also 3 cell. This is 1, this is second, and this is egg cell, right? So egg apparatus having total 3 cell, and we can say there is no any cell wall surround this polar nuclei, and this is wall 1 cell. So this is wall 1 cell wall, we can say, or cell. So how many cell are there? You can count. This is 3 and this is 3, and this is wall means 1. So total we will be having 7 cell, right? And how many nucleus are there? You can see here 1 2 3, this is 4 5, and this is 6 7 and 8. So nucleus are 8, but cells are 7. So it is 8 nucleated and 7 cell angiosperm embryonic sac. So most of the embryonic sac in angiosperm are 8 nucleated and 7 cell. So you can read this egg apparatus, antipodals and polar nuclei, right? And in case of double fertilization, we are going to see about what is role of this, right? Like what is function of this synergy, as I told you earlier, although we will be having look on this in next lecture, double fertilization, and what is function of this polar nuclei, right? What will gonna form from it, right? So, and some little things. So what is, now you can see what is the ploidy of the cell of this nucellus mmc. You can see nucellus have ploidy, it is 2n, right? And because you know from this nucellus it is mmc it is forming, so mmc has also 2n diploid, and from this diploid, by the, you know, which process, there will be a meiotic process, and we will be having only functional megaspore. So actually we had a 4 tetrad, right? So 3 of that 4 had already degenerate and one remain functional. That's the reason we will be having ploidy of this functional megaspore, it is haploid. And from this functional megaspore, all female gametophyte, or you can say embryonic sac, it is developing. That's the reason female gametophyte has also, you know, ploidy of haploid nucleus, right? So I hope this video may be helpful to you. If you like this video, so kindly subscribe this channel and share with your friends. Thank you.